Hello, my name is Alexa Grace Baldwin, and I am proposing a study to create a model organism from the mutated CFTR gene found in cystic fibrosis patients. Cystic fibrosis is an autosomal recessive genetic disorder that affects approximately 30,000 people in the United States alone. Cystic fibrosis can cause multiple pleiotropic effects, including bacterial infections in the lungs, mucus buildup, and inflammation, and many of these symptoms lead to lung cancer, which is the leading cause of death in cystic fibrosis patients. There have been great strides in the treatment for cystic fibrosis, but research is still in demand because there is still no known cure for CF. Therefore, in this experiment, I will obtain a DNA sample from a cystic fibrosis patient, isolate, amplify, and then clone that into a bacterial cell, E. coli. So, a little history of cystic fibrosis. It was discovered in 1938 as a separate disease from celiac disease. It was thought to then be caused from improper mucus secretions, but then in the 1950s, the discovery of the sweat electrolyte defect showed that mucus secretion was not the only cause of death. And then in 1989, the field of cystic fibrosis research was tremendously changed after the discovery of the CFTR gene, which is the mutated gene in cystic fibrosis patients. This discovery created a new area of treatment for cystic fibrosis patients called gene therapy. And due to these discoveries and gene therapy, life expectancy for CF patients is now 30 years, with many patients living to be in their 40s and 50s. And although there is no known cure, gene therapy has made remarkable improvement in the health of these patients. And gene therapy, it just involves transforming these corrected copies of the CFTR gene into the mutated copies through epithelial cells. The airway epithelium of CF patients has been the center of focus for the gene transfer. These therapies provide a substantial improvement rather than targeted specific symptoms of the CF patients. And although gene therapy has made great progress, improvements in this technique are still in demand. Different patients have different mutations, different symptoms, and many of these mutations cannot be treated with just gene therapy. Therefore, I'm creating a gene model. This model will be used in further gene therapy research to create new therapy methods, clinical trials, and for other research opportunities. And because gene therapy is a developing science, creating this model is very significant to cystic fibrosis research. This bacterial cell can be used for creating new medicine and evaluating various gene therapy strategies, and ultimately increasing the survival rate for cystic fibrosis patients. My procedure is broken down into 3 main steps: DNA isolation, DNA amplification, and then cloning. I will first collect a DNA sample from a CF patient, and I can use soap lysis to open up these cells. Then, using my Clone Jet PCR kit from Fisher brand, I insert the CFTR gene into a plasmid, and then using enzymes provided in the PCR cloning kit, the plasmid and gene are digested. Both the restriction enzyme and ligase are provided in this kit. The bacteria I'm using is E. coli. The heat shock, it's an incubation tube at 55 degrees Celsius in a hot water bath. The heat shock opens up the bacteria, allowing it to take up the plasma. In conclusion, my research project will be creating a model organism of a CFTR gene. Gene models are excellent research tools that are non-invasive, observable, and repeatable. This model will allow for better understanding of the exact functions of the CFTR gene. This research can lead to groundbreaking medicine and gene therapy treatments, and ultimately lead to the cure for cystic fibrosis.